This podcast is for informational and educational purposes only and is not to be considered medical advice for any particular patient. Clinicians must rely on their own informed clinical judgments when making recommendations for their patients. Patients in need of medical advice should consult their personal health care provider. From UPMC Children's Hospital of Pittsburgh, welcome to That's Pediatrics. I'm Steph Dewar, one of your hosts. I'm Vice Chair of Clinical Affairs and Pediatric Residency Program Director and a member of the Pediatric Hospitalist Division.
And I'm John Williams, Chief of Pediatric Infectious Diseases, and I'm delighted to welcome our guest today, Dr. Melissa Kane. Dr. Kane is an Assistant Professor of Pediatrics in the Division of Infectious Diseases and a member of the Center for Microbial Pathogenesis. And the central focus of her research is to understand the genetic and immunological basis for protective antiviral immune responses, as well as to study retroviruses. Dr. Kane, welcome.
Thank you.
So let me just start right off with a kind of basic question, but for our educated colleagues listening to the podcast, can you hum a few bars about retroviruses for us? What are retroviruses?
So retroviruses are a family of viruses that are special in that the viral particles contain RNA, and then upon entry into the cell, the viral enzyme reverse transcriptase copies that RNA into DNA that is then imported into the nucleus and permanently integrates into the host cell of the infected individual. So I know people say that herpes viruses are forever, but retroviruses really, really are forever, and that we have some in our genomes that have been with us for millions of years.
So you mentioned this. This is really intriguing. I think everybody's aware of the very, very important human retrovirus, HIV, the cause of AIDS. But you mentioned there are other retroviruses that are just in us right now.
Yeah, absolutely. So what happens when retroviruses integrate into the genome of an adult individual is that all those cells in every daughter cell is infected. But if a retrovirus happens to integrate into a cell that becomes a sperm or an egg that then becomes a new individual, all of the cells in that new individual are going to carry that virus with them. And of course, this can only happen if that doesn't have a huge detrimental effect on that new person. And that's why most of the retroviruses that we carry around in our genome are actually benign and not doing much of anything. The other thing that can happen, though, is that they can actually be beneficial. And we were just discussing earlier that a great example of this is we have an endogenous retrovirus that allows us to have placentas. So the retrovirus envelope gene causes the cells to fuse together in the placenta, allowing this multinucleated organ to exist as a barrier and conduit between the mother and the fetus.
And that's good because we're pediatricians. So not only did we all come from placentas, we like babies, we like placentas. Are there other good things that retroviruses do?
Yes, my personal favorite endogenous retrovirus is the one that allows us to have salivary amylase. And what that is an enzyme that allows us to start digesting sugars in our mouths. And this is something that only humans can do among the primates. And this is because of an endogenous retrovirus that we have, and even our closest relatives, the chimpanzees, do not.
So could you tell us a little bit about what you're working on now, where your research is going, or what questions you're asking?
Yes, absolutely. So the question that I first became fascinated with concerning retroviruses was why some people get infected and don't develop disease, whereas most people do develop disease. So the most common example of this is, of course, with HIV infection. Everybody knows you really don't want to get that, but what many people are actually not familiar with is that a small percentage of infected individuals actually control the virus all on their own without any drugs. And we know that it's their immune response that's doing that, but we don't really know the features of it that allow them to do that. I'm a molecular virologist and a geneticist, but I don't actually study human genetics. I use mice as a tool to ask these questions. So just like humans, mice have retroviruses that infect them naturally, and mice have genetic differences that also affect their ability to control infections, like their mouse retroviruses. So we use mouse models of a leukemia virus and a mammary tumor inducing virus. So marine leukemia virus and mouse mammary tumor virus. And we have all these different mice that do or do not control these infections. And we're asking questions about the genes that allow them to control these infections and the features of the immune response that make them able to do that. So what kinds of antibodies do they make? What kinds of cytotoxic T cells do they make? And on the flip side, I'm also interested in how individual cells try and control infection. And this is what we call the intrinsic immune system. And that's where I can actually ask questions about the retrovirus that we all really care about, HIV-1. So when the virus infects a cell, the cell has all sorts of tools that it tries to use to stop the virus from infecting. And I'm particularly interested in one called MX2. And it basically sits at the nuclear pore and tries to stop the virus from getting into the nucleus. And we're trying to find out how that functions at a molecular level.
So is this leading towards potentially a vaccine, or is it about modulating people's immune responses, or both?
Well, both, but that's also what vaccines do, right? So if we can understand how some individuals can make a good immune response against these viruses, we can potentially induce that immune response or prepare people to make that immune response before they're even exposed to the virus. So yes, the end goal is certainly a vaccine. With the more molecular stuff, it would be perhaps targeted drug development that can directly inhibit specific parts of the viral life cycle. But the questions that I'm asking in the mouse models is really to try and understand the pathways that allow for the production of protective antiviral immune responses so that we can try and recreate those in people.
So you mentioned that there's all these steps the virus has to go through to get into the host cell, get RNA turned into DNA, and then get into the nucleus. I imagine these are all steps where one could intervene with drugs or therapeutic molecules.
Yes, absolutely. And in fact, the drug cocktail that individuals who are infected right now take targets the virus at those specific steps. So because our cells don't have a reverse transcriptase gene, that can be a target of a drug. And because our cells don't have an integrase gene that's going to insert DNA into new places, we have drugs that target that. However, as we all know, these drugs are acceptable now for maintaining people who are infected for a long time, but we can do better and we can keep improving the side effects. The other goal is actually to develop drugs that are preventative. So one of the things that's happening in the field right now is trying to use these anti-retroviral drugs as a pre-exposure treatment while we're trying to develop a vaccine. So for high-risk individuals, they can take these medications to prevent infection in the first place.
So where did you get your energy or passion around these viruses, and what led you to these questions?
So as an undergraduate, I studied animal science because I was pretty sure I wanted to be an equine veterinarian. And after I took all of the required courses for vet school, I was looking for other science electives to take. And I took both an immunology class and a virology class. And I immediately became fascinated with viruses and how these little packets of protein and nucleic acid could do so much damage and take over our cells so effectively. And then I got into vet school and realized I didn't really want to go and had a crisis of trying to decide what to do. And a professor that I had become quite close to as an undergrad, Dr. Bruce Curry, encouraged me to try to get a job as a research technician because he thought I would love science. And it took me a long time because I had no research experience as an undergraduate because I hadn't considered it as a career. And so I ended up getting a position at a laboratory at the National Jewish and Medical Research Center and at the University of Colorado at Denver in the laboratory of Dr. Roberta Polanda, who studies B cell development and the production of autoreactive B cells. And I immediately fell in love with science. And then when I went to graduate school at the University of Chicago, this very, very serious Russian professor, Tanya Golovkina, came to me and said, I picked your application to come here, come rotate in my lab. And that was where my love affair with retroviruses in particular began.
It's really remarkable. So many, for an individual scientist like you, right? what an impact a few meetings with mentors, with individuals who inspired you with particular scientific things, what an impact that's had on you.
Yeah, absolutely.
And it's brought you into a field where similarly, when Howard Temin and David Bolton discovered reverse transcriptase, there was no knowledge about HIV then. There weren't human retroviruses. These were basic molecular virologists trying to understand basic science. And yet if those sort of coincidences, you might say, hadn't happened, boy, the world would have been in trouble when HIV emerged.
This is a great opportunity for me to plug basic science. So I attend the Cold Spring Harbor Retroviruses meeting every year. I skipped last year in setting up the lab, but I won't miss another. And one of a longstanding member of the retrovirology community, John Coffin, said, I don't remember which year this was, but he said how important it is to remember that if people had not been studying these viruses that no one cared about when HIV came on the scene, it would have been at least another decade before we had any treatments. And it really is thanks to people who were studying chicken viruses and mouse viruses and horse viruses that we have the medications that we have for HIV and that they were developed as quickly as they were. So I just like to give a little plug for people studying things that may not have an immediate translational impact.
Well, I think it's a common experience that especially non-scientists, even if they're physicians or other people in the health field, often have is, boy, why are you people studying fruit flies? What can we learn from fruit flies? And I think, yes, it's very important to study a particular disease, but you can't always predict what's going to be important.
Yes, absolutely.
Or what you're going to find. I mean, we've had this conversation with previous guests on this podcast about both of these issues, the influence of individuals in their career, and also just discoveries. I can think of the conversation that we had about diabetes and our pediatric surgery colleague about the unexpected finding that they had. So it's just fascinating. So that's how you came to the viruses. How did you end up here at UPMC Children's Hospital in Pittsburgh?
That is thanks to Dr. Carolyn Coyne. So I actually was on the job market for some time, searching for faculty positions and interviewed a number of places. And then last spring, when I was out interviewing other places and actually had an offer from another institution, this job posting appeared that I brushed aside because I assumed there wasn't time for it to get processed. And my postdoctoral mentor, Paul Beanosh, came up to me while I was splitting cells in the lab and said, I think you should apply to this job. I said, well, there's no way, there's no time. And he said, no, I'll email Carolyn. And then she brought me out here rather quickly for an interview and to meet everyone and to see a little bit of the city. And the rest, I suppose, is history, that it just turned out to be the perfect fit.
What about this city or this location or this environment for your particular research interests are particularly helpful to you?
So I think what's really exciting about this place for me is that I'm not a clinician, but I do think it's important to be reminded of the downstream impacts of what we do here. So even just walking over to this building today, I'm walking by a number of patients and their families. And sometimes if you're having a day where you're not feeling particularly motivated, reminding yourself that there are real people hoping that you're going to take this work seriously and move it forward can really get you out of bed in the morning. The other thing is that I wanted to be in an actual city. I'm a city girl where you don't have to drive a car. I don't have a driver's license. And Pittsburgh is just big enough and just small enough for that to work for me. And also there's just a number of really wonderful virologists here. So it's great to be a part of that community.
Yeah, we're lucky to have a really good community. And I agree, Pittsburgh's like the Goldilocks size. So we should thank Carolyn Coyne for bringing you here, although can we plug our fellow podcast host? Is that?
I guess we can give her a shout out on our episode.
Okay, but Brian Martin had nothing to do with recruiting Dr. Kane here. And I think too, the point you made about how basic science can lead to advances in clinical care and, walking past the Children's Hospital. So when Dr. Dewar and I were young doctors, which was a few years ago, pediatric HIV/AIDS was huge. Lots of kids around the country, even in not big cities, were being born with HIV and developing AIDS and dying very young because we just didn't have good drugs. And because of the work of scientists like you, not only did we get good drugs and figure out how to use them, but people figured out how to prevent transmission to babies. So almost no babies now, should be born with HIV/AIDS and have to go through what we saw. So I think for us, it's a very tangible example of exactly what we're talking about, is how basic science can really make a dramatic difference, even during, one individual's career.
Just another example of how science has changed our clinical care. That is just so remarkable, because that was a devastating clinical.
And it really wasn't that long ago.
No, it wasn't.
No, we're not that old. It was not that long ago.
It doesn't seem that long ago.
It was not.
No, it wasn't. But I mean, it was. We had no hope to offer them, and it was devastating.
In fact, when I was a medical student, I wasn't sure I was going to do infectious diseases, but I liked pediatrics and I liked infectious diseases. And people then told me, if you're going to do pediatric ID, you are going to be an AIDSologist. That's all you'll do, because that's what so much of it was then. And now it's so uncommon that I'm barely competent to take care of a child with HIV, because you need somebody with more expertise, and we just don't see it much. That's a wonderful thing.
That's a real testament to the science.
Well, Dr. Kane, I want to thank you so much for coming to visit us and sharing your research with us.
Yeah, absolutely. This was fun.
Yeah, and I'm sorry about the experience that you had in vet school, but I'm happy that you rebounded.
Oh, no, I never went, so it worked out fine.
Their loss is our gain.
Certainly. You can find other episodes of That's Pediatrics on iTunes, Google Play Music, and YouTube. Be sure to subscribe to keep up with new content, and leave a review and tell us what other topics you'd like our experts to cover. Thanks for listening.